on.
Yes.
Okay. Sorry for making you wait. Maybe you're a few minutes late.
No, those were great talks.
Yeah, that's it. And these are talks where you have so many questions. Okay, great. So Professor Muotri is at the Department of Pediatrics and Cellular and Molecular Medicine at UC San Diego, and is director of the Stem Cell Program and Archaealization Center. I have no idea what archaealization means, but it sounds very impressive. I have to say that UC San Diego has been at the vanguard of neuroscience and incidentally also of artificial neural networks for a long time, and still one of the main centers of research, especially at the intersection. If you look at the intersection of neuroscience and stem cell biology, which is where Professor Muotri works, well, there's very few places in the world that have both that School of Neuroscience and the Salk Institute next door. Anyway, the podium is yours.
All right, thank you. Get started here. Yeah, so I'll be talking about the work in my lab with brain organoids, and in a moment I'll introduce you to brain organoids. But just before I started, I just would like to challenge you guys, the audience, to think, to pause and think about the major problems that we are facing in the world, or we're going to face in the future. And if you are like me, most likely you thought about one of those things. So these are very challenge problems that we have. But one thing is for sure, there is only one solution for all these problems. And the only way to solve it is to use the human brain, especially your cortex. That's your cognition power here. So the problem with that, is that we don't fully understand how the brain works or how the brain is formed. And I choose this image to illustrate better what's happening. So if you look at those images, you know, all these pieces here, you can easily recognize that this is a car. So if I'm to break the brain in all these different pieces, not even a neurosurgeon can actually tell that it's a brain or be able to reconstruct the brain from scratch. And that's because we don't know how to make it. So if you don't know how to make it, you don't know how it works. And the major bottleneck to find out how the human brain works, it's because it happens in uterus. So you don't have tools that are powerful enough to really follow brain development, such as looking for the first synapses, the first neurons to fire, the first networks to form. So these happens all inside the womb, and you might have like ultrasound that doesn't have the magnification enough or the power enough to really focus on the synapses or the neurons. So because of that, the human brain, how it's formed in the first 9 months, it's actually a black box. We don't know how it does. We have to rely on model systems that are not always perfect, such as the mouse brain. The mouse brain is actually formed in 20 days. So you can see how different it is, the mouse brain from the human brain. So that's why we rely on brain organoids. Brain organoids are three-dimensional structures that you create from stem cells. And these stem cells can be derived from human embryonic stem cells or induced pluripotent stem cells that can be generated from a live person. So I can take a skin cell, I can reprogram these skin cells to become induced pluripotent stem cells. And from that, I can make a brain organoids, which is recapitulation of the neurodevelopment outside the uterus, inside the lab. Brain organoids are not mini brains. So this is the term that media uses to refer to this technology, but conveys the wrong idea that you have a miniaturized brain in the lab inside a dish. And that's not true. There is actually lots of limitations of this technology. For example, it's not vascularized. So that's why it grows to only to 0.5 centimeter and not more than that. It's actually not a representation of the entire brain. We don't have all the pieces there. It's also not connected to a human. So because of that, we don't like to call them mini brains. So this is actually how they look like. This is Isabella, is a postdoc in the lab. She's handling a plate with some of these organoids. And you can see these white dots, each one of them is one of these organoids. So we actually grow in the lab thousands and thousands of these brain organoids from different people, different individuals, most people with neurological disorders. And these organoids, if you cross-section one of them, you see that they have this nice structure that resemble the human fetal brain with a small ventricular zone and in the cortical plate surrounding that. So it actually has a very nice anatomical definition. And the cell types, although we don't have the same number of cells in the brain, we have about 2.5 million neurons in there, which is equivalent of a bee brain. So it's a very tiny thing. But we do have the cell types that are major components of the brain, such as neuroprogenitor cells, excitatory neurons, inhibitory neurons, and glia cells. We have all the pieces to form networks. So we use these organoids to understand how the brain is formed, as well as what kind of insults can damage the brain. And one example is our work with the Zika virus. By exposing these organoids to the Zika virus, we show how the microcephalic brain in the affected babies was happening. And more importantly, once we know how that happens, we can screen and design drugs that can block the action of the virus. So just 2 years after the outbreak, we were able to find a drug that can actually protect the infected mom from having a baby that's infected with the Zika virus. We are doing the same thing for the coronavirus. This is an article from the New York Times reporting about the neurological impact of the coronavirus. And our work with brain organoids is helping to explain why. Why do we have this neurological component in this pandemic? But one question that for several years remains kind of a challenge was to generate brain organoids with functional networks. Most of the protocols that we had in the past, they could not generate organoids that actually function like the brain. So that was a major challenge, and we took that to the heart. So we optimized this protocol to create a brain organoid that will actually fire. And we show that by placing these organoids directly on a multi-electrode array. So these arrays are electrodes printed in the bottom of a dish that capture the electrical activity, so you can measure that. The beauty of that is that you can do it longitudinal. So you can see how the networks evolve. And just to show you an idea, so these are a mean firing rate, an overall brain activity, and you can see that the primate brain will have like something about 20 hertz. And most of the in vitro work is here below 5 hertz. So our organoid is in here in this line here. You can see that starting at 25 weeks, it kind of almost exponentially grows in terms of activity, reaching something close to this 20 hertz when they are about 40 weeks or 9 months of age. So that was really unprecedented in tissue culture. Never before a human-made brain network could reach that level of activity. And then we decided to see if we can generate these neural oscillations similar to what you can capture with EEG or other electrodes that you place close to the brain. The reason why this is important is that we can cross the bridge between the basic biology and cognition, if we can do that. So we decided to check if there are these neural oscillations coming from the brain organoids, and they actually do exist. They start when they are about 4 months of age, and they become highly synchronized when they are 6 months of age, and highly complex when they become 8 months of age or longer. And if you stay longer enough, I mean, they actually generate quite complex different frequencies of oscillations coming from these organoids that actually matches human neurodevelopment. This is what we predicted using a machine learning algorithm to compare the electrical signal from an organoid with the EEG from human brains. And starting at 25 weeks, I mean, there's a perfect correlation suggesting that the same trajectory of network is being formed in these organoids as is formed in the human brain. So that was a big jump in this technology. So before that, as I said, we could use the morphology of the organoid, but not the function of the organoid. So that created a big buzz in the scientific world, because now we have a tissue that we create and behaves like the human brain. So I like the title of this New York Times article saying that organoids are not brains. How are they making these brain waves? That challenges the concept of what we can have in vitro. And of course, I mean, it touches on the ethical concerns about the creation of these organoids. But nonetheless, I mean, we are using these organoids now for several things. For example, to discover new medications or new treatments for different neurological disorders. This is a list of genetic disorders that we currently have now in the lab. We're creating organoids for each one of them and using them to test drugs or to test gene therapy so we can enter a clinical trial for these conditions. Another thing that we can do is to stimulate maturation of these organoids by adding input and output. We do that trying to mimic the human biology. So one way to do it is to add a retina, man-made retina that connects to the cortex. So what we have here is the retina fused with the human cortex. So you can see the photoreceptors in green and the projections in red. So the hope is that we can mature these combined organoids now so they can receive visual stimulation and record memories in the cortex. So we're not quite there yet, but this is something that we're working on. Another strategy that we have is to embodiment of these organoids with a machine interface. So what you are watching now is a robot that is learning how to walk using the electrical activity coming from one of these organoids that are now sitting inside an incubator. So it sends the signal wireless to the robot while it's teaching how to move the four legs. We are now adding sensors, such as these two eyes that the robot has. So these are actually infrared sensors that informs the organoid when the robot is approaching an obstacle. And by doing that, it stimulated the organoids to create a new response that drives the robot in another direction. So we are doing that with a partnership with Microsoft to actually inform better artificial intelligence algorithms so they can become cognitively more similar to the human brain. Another use for these organoids is to actually understand how the human brain evolved. You probably know that there were different types of humans on Earth, and we know more about the Neanderthals because they actually overlap with us. But even the Neanderthals, they don't have the sophistication, the technological sophistication as the modern human brain. So this is just one idea to illustrate what I mean. This is the time frame that the Neanderthals explored the Earth, and you can see that they developed some tools. But we, in a very short period of time, we were able to create a very dramatic environment to change the technology that we have around us and create all kind of new tools that you can imagine. So this is very different from any other species, including the Neanderthals. But what makes the modern human brain very unique? Unfortunately, we cannot create organoids from the Neanderthals because there is no soft tissue, no live cells to reprogram and create these organoids. But we can take a genomic approach. And that's what we did. We compared the genomes of the Neanderthals with modern humans, and we end up with a catalog of genes that are different between us and them. And then we use genome editing technology to archaealize the genomes of modern humans, creating brain organoids carrying ancestral genetic variants. And one of the interesting observations that we have is that a single gene was able to dramatically change how neurodevelopment happens. So this is just one example of the electrical activity on the archaealized organoid compared to modern humans. So you see that they have more activity very early on, and then they decrease the number of synchronization of these networks. This is exactly or very similar to what happens with our close relatives, the chimpanzees. So they have this fast neurodevelopment. To be honest, a baby chimpanzee can actually outsmart a human baby. But later on, I mean, we're able not only to catch up, but to increase our cognitive abilities. So it seems like these kind of genes are doing something very similar here. So that's one application. We are also thinking about the future. How can we take this technology to the future? And we are using genome editing to accelerate human evolution and create new networks that were never present in evolution before, perhaps networks that would be more resistant to diseases such as Alzheimer's or dementia, or networks that would be even smarter than the networks that we have right now. So these are some of the projects that actually make me think about how we want to shape our future. And one idea we had was to send organoids to the space station so they can grow in microgravity. There are a couple of missions where we send these organoids to live and develop in that environment. And this was inspired by the NASA twins experiment, where they analyzed the brother that stayed in the space station for 1 year and compared to his brother, genetically identical brother that actually stayed here on Earth. And basically, microgravity affects different systems in your body, creating something that it's similar to an accelerated aging after a while, including cognitive decline. So it's interest for NASA in future space colonization to understand how to mitigate the environmental insults coming from space. So that's why we are sending these organoids there. So the astronaut basically receives these organoids inside a box that is fully autonomous. And the only thing that they have to do is to place this box inside one of these cabinets, so we have immediately access here on Earth, and we can follow up the development of these organoids. And one thing that we expect to see is a recapitulation of this accelerated aging. So this will not only help NASA with future interplanetary colonization or long distance travels, but we also think that this could be a model for late onset neurological disorders. As I mentioned, Alzheimer's, Parkinson, dementia. These are diseases that takes a very long time to develop in humans, and we simply don't have good models for them. So by able to age organoids in space, we might be able to take advantage of what space can do to your brain to find better treatments and cures for neurological disorders here on Earth. So there are many other applications that I decided not to talk, but I invite you all to visit our stem cell channel. So this is a house here at the UCSD stem cell program. You can see different videos in here about how we are using stem cells and brain organoids to solve many questions in biology and engineer. And I'll stop here by thanking members of my lab who actually participate in this research and my funding agencies. Thank you very much.
Wow, breathtaking. Wow, I knew it was going to be interesting. I had no idea there was so much. Okay, you have a question in the QA, 2 questions in the QA, one from Kurt, one from Mark, 2 from Mark. Can you see them?
Is in the Q&A?
Yes. Kurt is asking, how long do the organoids remain viable? What is the evidence for their degeneration?
Yeah, we were able to keep the organoids for 3 years, and then we just stopped it. And we stopped it because we didn't want to keep them alive because we didn't have a use for them. After 3 years, they stopped growing. The neurons inside the organoids kind of die. This is a process that is kind of a necrosis, similar to necrosis, because they stop receiving nutrients coming from the environment. So as I mentioned, they are not vascularized. This is something that we're working now to vascularize these organoids so they can be alive for longer periods of time. But nonetheless, the neurons that surround the organoids, as long as you keep feeding them, they would be alive, pretty much as the neurons in your brain. I mean, that they were born when you were born, and they stay with you for the rest of your life. So as long as you have nutrients, they are alive. And what is the evidence for the neurodegeneration? Yes, we can see the neurodegeneration inside the organoids. And then for Mark, can organoids be networked together and specialized? Yes, we are working in a project like that. We are creating like a super net of organoids that communicate with each other and actually learn with each other to resolve a specific task. So this is ongoing. We have preliminary evidence that this might be the case, but we haven't proved it yet.
There's one more from Mark. Do they
respond to the analog of a reward? Yes, that's exactly what we are trying to measure now. Yeah, we think so. But again, there are some experimental issues to actually prove that this is the case. But we hope to have that by next year.
Are there more?
David,
have
you been able to induce epileptic form activity in organoids using typically epileptogenic interventions? Yeah, you can stimulate these organoids with some drugs that actually induces some kind of epileptic form. We also work with genetic forms of epilepsy, early onset of genetic forms of epilepsy, such as CDKL5. It's a gene that causes early epilepsy in children, and we can see alterations in the networks. So we are using now the system to actually screen to produce better drugs or treatment for this population. I have one more here. We'll be able to implant organoids in human brains to compensate for damage, such as stroke, MS, or other brain damage. Yeah, that's one of the applications that I haven't mentioned. We can actually tailor a specific organoid to a brain region that is missing, or to produce neurons or specific neurotransmitters that are important in certain diseases. For example, in Parkinson, we can create a small striatum that produces a dopamine and transplant back into the human brain. We're also working with TBI, traumatic brain injury, to replace specific neurons that happens in some of the cases. Yeah, you can tailor the organoid to specific brain regions.
How big is your lab? Curiosity.
We are about 25 people.
Wow. I saw your website. It's impressive that 25 people can do so many things. And who else in the world is working on these things?
Oh, there is a boom now. I mean, most of the stem cell biologists that are interested on neurological disorders are actually moving into this field, and we are seeing like a growth in improvements of protocols. There's a new protocol every week. People are learning how to make them work better, to survive better. As I mentioned, I mean, there are several people working on vascularization. Yeah, so I think we are seeing like a exponential growth of the labs that can actually do this technology.
Is there a conference on brain organoids? Where do I go to hear the latest?
Yeah, Cold Spring Harbor has a brain organoid conference every 2 years. And then, I mean, if you go to stem cell meetings, you're going to see brain organoid works being presented as well.
Okay, fantastic. Thank you very much.